So we are going to move to the next presentation. Our next speaker will be Belinda Hernández. Belinda is currently the Senior Research Fellow, TILDA, at Trinity College Dublin in Ireland. She obtained her PhD degree at the University College Dublin. And her areas of research interest are mainly at the advanced statistical and machine learning methods to develop physiological and epigenetic biomarkers for health aging and multi-morbidity prevention. Let's welcome Dr. Hernández.
Thanks a million. Yes, so I'm here to tell you a little bit about the TILDA study. So I'll give you a quick overview of our study, then go through some of the biomarkers that we collect before showing some recent research from our group. So we are TILDA, which is the Irish Longitudinal Study in Aging. We've been going a little over 13 years at the moment. So when we started out, we had 8,500 participants, and we were nationally representative of 1 in every 156 community dwelling adults aged 50 and over. So we're quite representative, and we now have just over 11,000 participants. So just to note that TILDA is part of a kind of wider global initiative. We're part of what is known as the HRS family of studies. So we have largely kind of harmonized data and health assessments across around 40 countries. So we started out in 2010. We've currently just finished our 6th wave of data collection. That finished a couple of weeks ago. We capture survey data every 2 years, which captures data on participant data on social self-reported health outcomes in economic and financial data as well. Our biomarkers are largely consigned to the health assessment, which takes place every 4 years. We've had 3 health assessments so far, and there we collect a number of biomarkers from kind of blood, hair, saliva, and stool samples. So we have microbiome data from there. So this is by no means an exhaustive list, but it's just to give you an idea of the most commonly used biomarkers that we collect in our sample sizes. So we collect cardiometabolic, cognitive, a wide array of inflammatory markers, kidney, hormonal data from our hair samples and micronutrients, and we have that over longitudinally over 4 years at the moment. And as I say, we're in the middle of processing our 3rd time point, which will be longitudinally over 12 years. With regard to other data, then we also have epigenetic data, telomere and metabolomic data over 4 years as well. So that brings us on to some work that we've been doing in epigenetics. So, I mean, just to start, we know that there's high inter-individual variability in the pace at which we age, and that's shown here by our 2 sets of identical twins. Obviously, they're the same age, but one set looks markedly older than the other. And we know that there's been an awful lot of research in kind of developing a measure which can encapsulate the aging process in a better way than just chronological age. And one of the ways that we can do this is through epigenetics, or DNA methylation is shown the most promise. So there's kind of been an explosion of research in recent years in developing what are known as epigenetic clocks. So obviously we know that around 1/3 of methylation levels at around 1/3 of our CPG sites affect or are affected by aging. So we can use these kind of penalized regression or small n large p machine learning methods to uncover a very small set of CPG sites which can very accurately predict age. So we can compare then the residuals of these algorithms. So basically, if your predicted age is less than your actual chronological age, well, then we say you've a negative age acceleration. If the opposite is true, then you're biologically older than your chronological age, and we say you've positive age acceleration. So there's been quite a lot of different types of clocks that have been developed. And one of the first things we wanted to do in TILDA was to compare them and see, well, what sort of what sort of aging outcomes are captured by each of these clocks, because they're all developed on different tissues, they're developed on different cohorts, different age groups, and are even developed on different outcomes. So we wanted to see, well, do these kind of the most popular clocks work on our cohort, which is mainly white European. So we compared 4 different clocks, the Horvath, Hannum, PhenoAge and GrimAge. The first 2 were known as the first generation clocks. They were developed on age as the outcome. PhenoAge and GrimAge were developed using mortality as the outcome. So we compared these for 9 different functional outcomes. So we can see in our minimally adjusted models, which adjusted for just age and sex, we could see that PhenoAge and GrimAge were associated with slower walking speed and higher odds of polypharmacy in our minimally adjusted models. When we then adjusted for things like your socioeconomic status over the life course and things like health and lifestyle behavioural factors, well, actually only GrimAge was associated with walking speed and polypharmacy. So then we went on to look at other outcomes like frailty. We could see very similar results. So GrimAge again was significantly associated with higher frailty in fully adjusted models, and it was PhenoAge which was marginally associated with cognitive impairment. Again, for mortality, we could see that GrimAge surprisingly was the only one that picked up on mortality in our cohort, despite the fact that PhenoAge was also trained on mortality. Long story short, I guess GrimAge was associated with 8 out of 9 of these functional outcomes in our minimally adjusted models, and it remained significantly associated with 4 out of the 9 of them for walking speed, polypharmacy, frailty and mortality in these fully adjusted models. So I guess GrimAge was the one that showed the most promise. It was the only one really that kind of captured a wide array of different functional outcomes in our cohort. So this is just a snapshot of some work that's been done by our group in this area, and I'm really the reason why I'm showing epigenetics here is because we have a lot of studies that are ongoing at the moment where we're developing different surrogates for different health and functional outcomes, but unfortunately we can't share the results here today. So I just wanted to note that this is part of a wider collaboration with the US and a number of different European cohorts. So if anybody has epigenetics data and will be interested in joining this collaboration, get in contact. We'd love to further that. So another thing I wanted to show from our group is something called the active stand test. So this is a part of our health assessment which is unique within the HRS family of studies. It's unique to TILDA, although Professor Tan has shown that she also has a very, very similar study. So in the active stand test, it's a very simple test. The participants come in, they lie down on a bed for 15 minutes and they relax. Then they're asked to stand up as quickly as possible. They remain standing for 3 minutes, and that's the entire test. So during this time, you can see that they're hooked up to a phonometer, and this measures loads of data about their peripheral hemodynamics, or the blood pressure, or the blood flow around the body. So we capture things in real time, like our heart rate, systolic, diastolic blood pressure, and other measurements like stroke volume, total peripheral resistance. So another thing that isn't in this picture, but that we also capture simultaneously, is oxygenated and deoxygenated blood flow to the brain using NIRS. So this is what the data looks like. You can see that that we have these kind of curves from 30 seconds before standing. I'm showing until 3 minutes after standing, and this vertical line here is where the participants stand. So just to give you a brief overview of what actually happens when you stand. So when you're lying down, your blood is kind of evenly dispersed throughout your body, and it's in line with your heart. When you stand up, what happens is gravity acts on us. Our blood, it pools in our feet and our lower extremities, and that kind of activates our baroreceptor reflex. So then our sympathetic nervous system gets activated. That's the part of our autonomic nervous system that's in charge of the fight or flight response. So you can see like immediately, you can see immediately after standing that we have this dramatic drop in blood pressure here. This is our systolic and diastolic blood pressure. When our sympathetic nervous system gets activated, well, we have this compensatory increase in heart rate. So our heart rate starts increasing as our blood pressure starts decreasing, and that's to kind of get our blood flowing around the body again. So we usually hit our lowest point, or our nadir, in the blood pressure around 10 seconds after standing, at which point the parasympathetic branch of the ANS takes over again, and our heart rate begins to drop, our blood pressure starts to recover, and our pointer doesn't work here, but then we usually start to recover from about 30 seconds. We really should have recovered by 60 seconds after standing. If we haven't, that's usually a sign that there's a bigger underlying problem. So with regards to the blood to our brain has its own defense, our own defense mechanism called cerebral autoregulation. So, but despite that fact, there are actually transient blips or drops in the blood pressure to the brain in response to standing. So we can see here, this is TSI, that's the percentage of oxygenated to total hemoglobin in the brain, and we can see that there's a drop of about 2% in TSI on average in the initial response to standing before we get this recovery. So this is again a snapshot of some work that's been done by our group in this area. So most of this literature and the wider literature in this area, it's really focused on just looking at one time point or at a snapshot or a summary of these curves. And I guess when I looked at this, I was kind of thinking, well, actually maybe looking at just one time point doesn't encapsulate all of the information about the autonomic response to standing. Where we have a huge loss of information. So what I wanted to do really was to use the entire curve and look at the entire response to standing over time and see, well, can that predict, maybe can that be an early biomarker for disease onset or for mortality? So I just wanted to show two recent pieces of work that we've done in this area. So the first one was focused on diabetes and looking at the impact of diabetes on peripheral and these cerebral hemodynamic responses, and looking at its association with cognitive impairment. So type 2 diabetes, it affects about 10% of older adults in Ireland, but its prevalence is increasing. So it's known that, that diabetes is associated with greater risk of poorer brain health outcomes in later life. And our hypothesis really was that, well, maybe it's impaired cerebral perfusion, in other words, like these kind of impaired blood flow to the brain upon standing might link diabetes to these brain health outcomes. So the first thing we wanted to do was look at the impact of diabetes on these signals and see, well, first of all, is there a difference in the recovery of these signals in diabetes? And then we wanted to see, well, are there associations with cognitive impairment, or do we see anything there? So what I'm showing here is the marginal effect of diabetes, pre-diabetes and no diabetes on each of these curves. So the coloured areas are showing the time points where diabetes was significantly different, and this is after adjustment for a number of different things like age, sex, education, and health and behavioural factors, and also medication usage, because obviously medications can impact these signals. So we adjusted as well for things like use of antihypertensive, statins and antidepressants, antipsychotics, things like this. So we can see that even after this adjustment, there were marked differences in those with diabetes. So we can see that in terms of the heart rate recovery, it was this kind of initial response around the peak, which was significantly different in diabetes. So we can see that there's this kind of blunted peak response in heart rate in those with diabetes. And this is a known feature of autonomic dysfunction. It's thought to be kind of an inhibition of reactivating the parasympathetic nervous system. So we do see this in diabetes. We also see kind of poorer blood pressure recovery. So yeah, I here we go. So we can see kind of poorer blood pressure recovery. With those with diabetes have lower blood pressure in general in their recovery period from 30 seconds all the way towards standing. And we can see again that with regards to stroke volume, there's this kind of inability to lower stroke volume in the recovery period to below the supine baseline. And this is actually a feature that we see in pretty much all disease states and mortality in TILDA. With regard to blood flow to the brain then, we saw that those with diabetes, they had higher drops in their percentage of oxygenated blood flow to the brain, and this persisted up to 3 minutes after standing. So we did see marked differences there. With regards to the link to cognitive impairment then, we saw that, well, those with diabetes and with larger drops in their TSI in this recovery period were associated with higher odds of cognitive impairment. So our measure of cognitive impairment here was a MoCA value of less than 23. So, I mean, we did see that diabetes was associated with this impaired response to standing, and we saw that diabetes and the impaired recovery of TSI was also associated with higher odds of cognitive impairment. And I mean, this was by no means a causal analysis, but I think it does hint that that kind of this poorer recovery and bigger drops in blood flow in oxygenated blood flow to the brain in this immediate response to standing. It might be one important mechanism that links diabetes to poorer brain health outcomes. So the next study I wanted to show was looking at these signals at baseline, and we wanted to see, well, actually, could these signals be used as biomarkers for mortality and for cardiovascular disease onset? So I just wanted to show here, this is the mean curve at baseline. So these are the baseline signals, and this is the mean curve for those who survived the intervening 11 years and those who died is in the blue line. So we can see that there are marked differences in the recovery and in these curves. So in particular, we can see that those who go on to die, they have higher mean heart rate. They have, again, this blunted peak response in heart rate and a slower blunted recovery as well. And we can see, again, this kind of lower recovery of systolic blood pressure, which we also saw in diabetes, and this failure again to drop our stroke volume index to much below its supine baseline in this recovery period. And these are features that we keep seeing again and again. So then we wanted to see, well, can we see differences not only in who went on to die, but also in what they died of? So we looked at different causes of death, and again we can see that those who died of other causes, they had very elevated heart rate in general, almost no peak, and just a very flat recovery. Now we do know that of those who were marked as other, over 1/3 died of respiratory illnesses, so that might explain this kind of elevated heart rate there. Again, those with who died of circulatory causes, they had this blunted peak heart rate response and kind of lower blood pressure. And again, we saw this same feature of stroke volume where it was higher in the recovery period. So we developed algorithms to see, well, after adjustment for all of these other factors are like frailty status and health and behavioural factors and medication usage, are these signals predictive of mortality and cause of mortality? And we found that they were. So I'm going to show the results here for the same thing that we did for cardiovascular disease. So we removed those with cardiovascular disease at baseline, and we said, well, in individuals who are otherwise healthy at baseline, can we use these signals to go on and predict who goes on to have cardiovascular disease within the next 12 years? So overall, we found that these 4 components here marked in green, they were the features which were predictive of cardiovascular disease over the next 12 years. So we found that those with who basically had higher heart rate and this blunted peak response, that was one of the features. The other signal which came out as predictive was this kind of failure to recover diastolic blood pressure from about 20 seconds onwards. And again, we saw this same kind of failure to lower stroke volume index from 20 seconds onwards. So these were the signals or the features which were predictive of 12-year incident CVD after adjustment for all of these other factors. So we found we could predict pretty okay. This was the accuracy on test data, which was completely separate from the algorithm development. We had an AUC of around 0.7 and okay sensitivity and specificity. But I think more than the accuracy, what's interesting here is that I also went and looked, well, it kind of occurred to me that maybe what I've done here is essentially develop a more complicated way of measuring things that are already measured in the clinic, like underlying heart rate and cuff measurements of our blood pressure. And another measurement that's commonly used is for orthostatic hypotension, which looks at the drop in blood pressure at 60 seconds after standing. So I also looked at those and wanted to see, well, are those predictive of these different health outcomes? And actually they weren't. So we looked at OH at 10-second intervals from 10 to 60 seconds, and we also looked at baseline heart rate and blood pressure, and we found that those markers on their own were not predictive of incident cardiovascular disease or mortality or cause of mortality. But when we looked at the entire curve, it was these features and these curves which were. So yeah, so that's kind of it. I suppose really what this kind of points at is maybe our initial hunch was correct, that maybe by looking at this entire curve rather than just recovery at one time point or at a very short interval, we can uncover more kind of subtle relationships which can act as kind of preclinical biomarkers for disease onset and mortality. So yeah, I'd just like to finish up by acknowledging our sponsors and for thanking our hosts and the organising committee for inviting me here today and for organising such a wonderful conference. So thank you.